Hello everyone. Now we are going to start with the concept of single cell sequencing. So far we have been looking into different types of DNA sequencing and RNA sequencing data. And in all those cases, the sequencing data was obtained directly from the tissue. So it was coming from a mixture of cells. But from now on, we are going to look into methods where the data will be collected from individual single cells. So the conventional methods, the particular type of sequencing data that we have seen so far, those are actually bulk sequencing data. So in these bulk sequencing data sets, what you observe or what you obtain is sequencing data from a pool of cells. So the DNA or the RNA that is our target of interest is actually captured from the tissue itself. So what you see that we are starting with an input material which is a mixture coming from a large number of cells. So in the end, the measurement that we get is also somewhat of an average of all the cells together. In contrast, when we talk about single cell analysis or single cell sequencing, then we can actually individually look into each of the single cells that are present inside the tissue. So in case of single cell analysis, it actually enables us to capture our target of interest, so be it DNA or RNA, from the individual cells. So what you observe is basically what you can obtain is measurement corresponding to each individual cells. So in case of bulk sequencing, you can think of an analogy. It's like a fruit salad in a bowl. So you have multiple different types of fruits that are actually all mixed together inside this bowl, and it can be even worse. So imagine that if you take all of these fruits together and convert it into a smoothie, then it will be very difficult to identify each of the fruits from that smoothie itself. So that is the sort of measurement that we get from bulk sequencing data. So that is an average measurement, and this is not actually sufficient for studying different types of heterogeneous systems. So for example, an early developmental system or a very complex tissue like brain, or maybe even cancerous tissue which is very much heterogeneous, it will be very difficult to study this kind of systems using bulk sequencing data. On the other hand, single cell sequencing actually can enable you to identify the different types of fruit that are present in the fruit bowl or the mixture. So you can easily identify which ones are apples, which ones are kiwi, which ones are oranges, and so on. And in fact, this actually helps us in resolving the heterogeneity that is present within the tissue. So in fact, if we are looking into a cancer tissue, then we can identify each different types of cancer cells, the lymphocytes or the fibroblast or the macrophage or the other types of immune cells that may be also present in the cancer tissue. So the single cell sequencing is actually ubiquitous in its application in different fields of biology. So for example, of course, cancer biology is one of the main field or one of the most prominent field where it will find the most prominent application, because cancer is highly heterogeneous. So when you look into cancer, it's better to resolve cancer at the level of the single cell. Similarly, embryology or the developmental biology can also benefit from single cell sequencing kind of analysis, because when the embryo develops from a single cell stage, then it's actually a very diverse system, and within a very short period of time, cells actually acquire different types of functions, or so they differentiate. So that's why single cell sequencing will be very helpful in studying this development, how cells evolve over time, and how one type of cell can actually give rise to different types of cells. Similarly, in the study of the immune system, the study of the brain, as I said, like brain is a very complex tissue. So what are the different types of neurons and different types of other types of cells that are present in the brain itself. So in order to study all these different types of cells, single cell sequencing will be very handy. So in short, it's becoming more and more prominent that single cell sequencing is the future. So people are more and more these days, they are trying to generate single cell sequencing data from the tissue itself. And in fact, this has been actually there has been tremendous impetus in terms of like collaborations between multiple labs. Single cell sequencing actually started around 2009. Like that's when the first single cell sequencing data was produced, but it actually became more popular around 2013 2014. So in fact in 2013 single cell DNA sequencing and single cell RNA sequencing, they were chosen as the method of the year by Nature Methods. So every year Nature Methods actually declares one of the biotechnological methods to be the method of the year. So that was single cell sequencing into 2013. And since then, single cell sequencing has been tremendously explored in multiple directions. In fact, there have been a lot of development. Multiple different consortiums have been started, like this Human Cell Atlas project. Their target is to actually build a reference map of all the different types of cells that are present in the human body. And this is HubMap project is another similar project from NIH. And then again, we have this Human Tumor Atlas Network project where researchers are coming together to develop a complete atlas of the different types of cells that are present in different types of cancers. Not only humans, but there have also been like efforts to develop atlases for other kinds of animals, such as Drosophila or mouse. So those are also underway. So all these different consortiums, you can imagine that so many labs are collaborating in these consortiums that we have a large collection of single cell data. And every day you'll find new datasets being generated and new datasets being deposited in public repositories. So in short, we are actually living in the era of single cells, and it will only increase over time. And in fact, in last year, 2019, single cell multimodal analysis or multimodal sequencing was chosen as the method of the year by Nature Methods. So in case of multimodal sequencing, you will see that instead of measuring just the DNA or the RNA or protein, people actually try to combine multiple measurements from the same single cell. So we'll later on we'll observe some particular computational methods that are developed for multimodal sequencing data as well. So this figure actually illustrates all the different types of measurements that can be obtained from single cell. So of course, our cell consists of the nucleus, and inside the nucleus there is DNA. So if you actually can capture the DNA corresponding to the single cell, then you get single cell DNA sequencing data. If you capture the repertoire of mRNAs that are present in the single cell, then you can get actually single cell RNA sequencing data. So you can also capture some other types of things, such as the cell surface proteins or the proteins that are present in the intracellular space. You can also try to obtain some other types of epigenomic data, such as the histone modifications or maybe the chromatin accessibility profile from the single cells, and so on. And in fact, more and more methods are nowadays coming up where you can, as I said earlier, that multimodal measurements can be also obtained from individual single cells. So while this particular figure actually gives you all the different types of sequencing that are possible, or all the different types of measurements that are possible from individual single cell, in fact the single cell RNA sequencing is the most prominent one. So it is also the most mature technology in some sense. So at the very beginning, we are first going to focus on single cell RNA sequencing. So just before this, we actually discussed a lot of different computational methods for analyzing bulk RNA sequencing data. So while some of those can be applicable to single cell RNA sequencing data as well, but a lot of different novel methods have also been developed for single cell RNA sequencing data. And in fact, you will see that with this new kind of data, which is much more high throughput, much more high resolution, you will see that there are novel computational problems that are also coming up. So we are going to explore them in more details. So one of the reasons that single cell sequencing, single cell RNA sequencing became so popular was because there was a tremendous increase in the throughput when these droplet-based single cell RNA sequencing methods were developed. So this is actually an overview of Dropsec single cell analysis. So it actually utilizes some sort of microfluidic chamber, and then oil droplets inside which barcoded beads are combined with cells. So each cell is actually encapsulated inside a droplet together with these barcoded beads. These are the barcoded beads and these are the different cells, and they will be captured inside an oil droplet. So the reaction actually happens inside each of this oil droplet. So the mRNA is captured by first cell lysis, and in fact in the end these droplets, these individual droplets, are also barcoded differently. So these droplets can be actually finally pulled together and sequenced together. So in that sense you are actually obtaining individual reads from individual single cells. All those reads are basically barcoded, and then they can be very high throughput. So in fact this had the highest throughput when it came out around 2015, and the costs were also reasonably cheaper. So the cost of sequencing a single cell was around $0.05 US. So after this development of Dropsec, 10x Genomics came up with a very similar technology where they actually, their methods or their particular technology is known as GEM, which is gel bead in emulsion. So again, these gel beads are actually barcoded. So in fact the 10x Genomics next sequencing method, their particular, that is how they are termed. So it actually can sample around 3.6 million barcodes to capture or to tag the cells individually. So again, these barcodes or the gems are actually combined with cells, enzymes inside the partitioning oil, and then that will actually generate these single cell gems. So each of these single cell gems, since this gel beads actually contains those barcodes, the unique barcodes, after the mRNA is captured, they are converted to cDNA through reverse transcription process, and this cDNA is also barcoded, and this barcoded cDNA can then be sequenced using standard sequencing method, the short sequencing method like Illumina. So that's how they can produce very high throughput RNA sequencing dataset where around 50,000, 100,000 cells can be sequenced at the same time, and their throughput is very high, and cost is also reasonable. So if you want to know more about this particular technology of how 10x Genomics actually sequences these cells, you can go to this link and watch a particular very small video of where they illustrate this process. So this figure over here actually shows you the scaling of the single cell RNA sequencing experiments over time. So as I said, like the very first sequencing data, single cell sequencing came up around 2009. So initially you see the number of single cells in those studies were very less, like 10, 20, something like that. So then came up, then more and more technologies were developed, like CellSeq, SmartSeq, SmartSeq 2, and so on. So around 2015, this Dropsec was developed. Dropsec and InDrop, these are all droplet-based sequencing. And around that time, the throughput became much higher. So more than 10,000 cells could be sequenced over time at the same time. And things have improved over even after that. 10x Genomics, people actually now can sequence more than 100,000 cells. And there have been also development of combinatorial indexing technique, which are very much high throughput, and using multiplexing they can actually generate a large number of barcodes at the same time, so a lot of cells can be tagged. So and these processes are evolving. So now we are at a point where the experiments are very high throughput and reasonably costly, so a lot of labs can actually afford this kind of sequencing. So they can actually generate single cell RNA sequencing data corresponding to whatever is their tissue of interest. So let us look into the overview of the single cell RNA sequencing analysis. So what kind of analysis can be done and what type of algorithm we are going to actually analyze. So again, this after the single cell suspension is created from the tissue, then these barcoded libraries are generated, and then sequencing is done using a standard Illumina machine to generate the sequences corresponding to individual single cells. Now, those individual sequences from individual cells, then they are aligned and transcript quantification is done, just very similar to bulk RNA sequencing data. So in fact, in this kind of alignment and quantification, the same algorithms can be used, or some of their modifications, little modifications for single cells. So in the end, what you get is a count matrix. So remember previously when we talked about bulk sequencing data, we used to talk about samples, replicates. So maybe we used to have like around 8 samples in a particular study. Whereas in case of single cell sequencing data, what you have is measurements from thousands of single cells. So you get this count matrix, which is cells by genes or genes by cells, however you want to denote it. So maybe 20,000 genes have been measured for 10,000 cells, something like this. And this will be the size of the count matrix. And after that, some sort of quality control checks have to be performed to filter the low quality cells or the low quality genes. And then other type of technical effects can be removed through this batch correction method. So there are some technical artifacts that may be present in the different single cell RNA sequencing data. So for that also different algorithms can be developed for the batch correction method. And here you see one term known as data integration. So this data integration is actually also talking about computational methods which can be utilized for combining knowledge across single cell data sets. So imagine like from the same tissue, different labs have developed different single cell RNA sequencing data set, maybe using different single cell sequencing protocol maybe. But since the underlying tissue is the same, so they should have similar biological variation. And in order to capture such biological variation across these different studies, different data integration techniques have to be developed, because essentially since the sequencing protocol is different or the sequencing batch is different, the reagents, the chemicals, all those things are different. So there could be some sort of variability, some technical variability that has to be removed before the data can be integrated. So in case of quality control, also it involves the removal of unwanted transcriptomes, and in fact they can be also removing, they can also involve removal of doublets. So again, doublets are artifacts where two cells fuse together somehow. So you have to identify them, otherwise they will represent a mixture and they can confound your analysis. And the dying cells are not actually producing transcripts in the at the same level as any other steady state cell, so they have to be removed somehow. And also the contaminations. And again, batch correction is all about removing the technical artifacts. So again, this can be due to experimental batch or sequencing technology, as I talked about it. So the main purpose of the quality control is to remove the low quality cells or the low quality genes. And in order to achieve this particular quality control, sometimes some known RNA molecules are also added in the experiment, during the experiment. So these are known as spike-ins. So these spike-ins are a few types of RNAs which are already known. So their sequences are also known and their quantity is also known. So they can be actually generated artificially, or they can be also generated from a pool of RNA from a very distantly related species. So these are actually used as internal controls. So when you generate this read count matrix, so of course some spike in reads will also be present in the cells, in the different cells. And in order to identify the cells which are low quality, we can actually take two different approaches. So of course there will be some reads which will not be mapped, so there will be some amount of unmapped reads, and there will also be some reads which will be mapped to these spike-in molecules, these spike-in RNA molecules, which are not really coming from the cell itself, but these are all known RNA molecules which are added as internal controls. Now in this particular figure, so you are seeing the percentage of unmapped reads represented as a function of number of mapped reads. So each dot here is a single cell, And also the cells are also plotted like the percentage of spike in reads as a function of number of mapped reads. So you will see that the cells which are actually showing either very large percentage of unmapped reads or large percentage of spike in reads, they are actually low quality cells, because if most of the reads are unmapped, that is of no use. And again, if most of the reads are also, or a large percentage of reads are actually mapping to the spike-ins, that is also really indication of noisy measurements. So those cells must be removed. So in the end, we want to only keep the cells which are actually shown in green dots, which are the higher quality cells. Now let us talk about the second part of our single cell RNA sequencing analysis. So remember, after batch correction, we have the single cell count matrix. And one of the very first analysis that can be done is to do some sort of dimension reduction, so that we can actually visualize the cells. So of course, each of these cells is a very high dimensional vectors, where each of the cells are for each of the cells around 20,000 25,000 genes have been measured. So that type of data is very difficult to visualize. So oftentimes people actually apply some sort of dimensionality reduction method, either linear or non-linear, and they try to represent or visualize the cells in 2D. So, and after that, they can also be used for clustering or trajectory analysis. So these are all different computational algorithms. These will all involve different computational methods. And in fact, this clustering and the trajectory analysis, these are actually answering two different types of biological questions. Clustering is aimed towards finding the different types of cells that are actually present in the tissue, whereas trajectory is trying to identify the temporal variation of the gene expression values over time. So trajectory analysis is trying to capture the temporal variation of the gene expression from the snapshot. So it is trying to capture the development of the cells. And after that, the cells can be annotated. So after we have the annotated profiles, multiple different types of analysis can be done. Some sort of statistical analysis can be done to identify the cells which are corresponding to healthy condition as compared to the disease condition, and then their association with certain phenotype can be measured. Also, of course, we can go for differential gene expression analysis, which is one of the major application of RNA sequencing data itself, and then we can look for the enrichment of set of genes in different pathways. We can also do some sort of network analysis. So for example, we can try to identify the interactions between cell-cell. So this kind of cell-cell interaction network is actually going to give us idea about how cells interact with each other in the tissue, how they communicate with each other, what kind of signaling pathway they actually participate in. And at the same time, we can look into the gene regulatory network. So the gene regulatory network is going to again give us idea about how a set of genes can be regulated by another set of genes, and so on. So all these different types of biological questions, in order to answer them, we have to develop new computational methods. So we are going to focus on these different types of computational methods. So in fact, there are two different kinds of methods that have been developed for single cell RNA sequencing data. So one type of method actually deals with this removal of technical artifacts or the noises, the quality controls. In fact, in the quality control situation, we'll see that the single cell RNA sequencing data is also very sparse. So in such cases, some imputation methods have been developed. So we are going to first look into some imputation methods. And if time permits, we can also look into some batch correction methods, but we are going to mostly focus on this second set of computational methods, which are about efficient clustering and trajectory analysis, as well as this sort of downstream analysis like gene regulatory network reconstruction and so on. So from the next lecture, we are going to start with first we are going to look into one type of quality control method, which is about imputation of very sparse single cell RNA sequencing data. Thank you.